And welcome to the final lecture, Transcription Translation. Transcription Translation is basically all about how a cell actually does its job. So, Transcription Translation is all about how we go from the genes, which are the blueprints for proteins, all the way to proteins, functional proteins, which are the things that actually do stuff. These do the things. Right? Certain proteins made at certain times activate certain other protein pathways, which do certain other things, which maybe synthesize lipids, synthesize carbohydrates, activate respiration, what have you. Proteins are the prime movers. So transcription and translation is all about how we go from genes to functional proteins. And the first step in transcription translation is making something that can get out of the nucleus, right? Because your genes are encoded on your DNA chromosomes. And a chromosome is going to have hundreds to thousands, even in some of them, 10,000s of genes. They are very long, and they cannot, right? A chromosome cannot leave the nucleus. And that's the problem there, because ribosomes, they are the site of protein production, and ribosomes are outside the nucleus. So we are going to need to make a copy of a gene and make it mobile so it can go to the ribosomes in the cytoplasm. Now, this lecture is called Transcription Translation. Step 1, copy a gene, is transcription. Right? If you are transcribing, the word transcribe literally means copy. If I copy a book in English, my copy will be in English, right? Photocopying doesn't change the language. I emphasize this because the word translation means translating to a different language. In our case, genes are the language of nucleic acids. Proteins are the language of amino acids. Step 1 is transcription, which means we're copying, we're not changing the language. So in step 1, we have DNA genes, and we do transcription in order to get mRNA. And mRNA is still the language of nucleic acids. So, step 1 transcription is keeping the same language, nucleic acids, but just making a copy. And that copy is one gene. Because in general, one gene equals one protein. So when we transcribe, we are producing the blueprint for an protein. And transcription is all about making RNA. And making RNA is a little different from making DNA. In replication, we discussed the fact that in eukaryotes, there are 7 different chemicals in there, 6 enzymes, and 1 binding protein, right? So we had helicase, topoisomerase, then we had primase, DNA polymerase II, DNA polymerase I, ligase, and then our single-stranded binding proteins. Those were all involved. Helicase opened the helixes. Topoisomerase prevented supercoiling. DNA polymerase III laid down nucleic acids. Primase allowed us to lay down those nucleic acids. It was a somewhat complicated system. Lucky for us, we've got the home shopping network of enzymes in order to accomplish all of this for just making mRNA, right? We don't have primase, we don't have helicase, we don't have topoisomerase, we don't have DNA polymerase III. No, we just have one enzyme that does the job of all of those other enzymes. And what is this magical enzyme called? Well, my friend, that is RNA Polymerase II. Right? Call now. Supplies are limited. Buy your RNA Polymerase II in order to replace 7 different replication chemicals just to make your RNA. Call now, and we will double your polymerase. But anyway, RNA Polymerase II, right? It uncoils the DNA. It uncoils the helix. Right? It exposes the template. It copies the gene into the protein, or into mRNA, and it recoils the helix behind it. RNA polymerase II is a pretty badass enzyme. Now, you'll notice I said template singular. In DNA replication, both strands were template strands. In transcription, there's only one template strand, right? And that template strand is the 3' to 5' DNA, right? Our non-template strand is the 5' to 3' strand. So our template strand is 3 to 5. That means our mRNA, right, our RNA transcript is going to be a 5' to 3' bit of RNA. And that means, welcome to that memorable restriction, RNA polymerase II can only make a 5' to 3' RNA strand. Right? So RNA polymerase II has that restriction. The good news is we just ignore the non-template strand. So we don't have to have extra molecular jiggery pokery to handle the strand that's 5 to 3 that would, so we just use the template strand 3 to 5 in order to make our transcript, right? Not too terrible there. So we'll start talking about how we get this process going in our next short. But I think just knowing that it's RNA polymerase II and that these are our restrictions is good enough for now. So see you in the next short on promoters.